Hi, good evening. This is your instructor, Dr. Monica Gullianas. Today I will be talking about the last portion of DNA damage and repair. So we have been talking about DNA damage and repair for a while now, but this is the last of this module. So we have already talked about a number of ways DNA can be damaged. It can be replication error, it can be a chemical or physical damage to nucleotide, it could be damage to the sugar and phosphate backbone of DNA. So the damage can occur in different ways. We talked about a number of ways the cell can help repair that damage as well. So now we will be talking about post-replicative repair, or more commonly known as double-stranded break repair. And as you all know, that double-stranded breaks are very, very hard to repair. These are less accurate and they're more likely to lead to mutation. There are three major mechanisms by which double-stranded DNA breaks are repaired: recombination, translesion, and non-homologous end joining. In recombination repair, this repair is usually the large bulky damage that has occurred to DNA is repaired by recombination. The example is again pyrimidine dimers that we have been talking about a lot, because these pyrimidine dimers are pretty common, especially in damage by radiations. And these damage can block DNA polymerase 3. But the replication fork can still move past these blocks and continue synthesis and leave these single-stranded DNA behind. And they can do this by using recombination. And here I will show you the example here. So in this example here, this is the parental strand, and these are the daughter strands, right? So this parental strand has a thymine dimer. This thymine dimer is it will block the replication because the daughter strand doesn't know what to add here. This thymine dimer doesn't really pair with anything, so it's hard to know what is it. So what the cell can do is it just leaves this portion here unreplicated and make a recombination. That here it recombines to copy this information from this part of the similar part of the DNA by looking for homology there. Once it's find the correct information, it will replicate this information here on the daughter strand, but the parental strand still have the damage there. So what the recombination repair does, it doesn't really correct the damage, but what it does, it buys some time for the cell to do it. So now the daughter strand is fine, the daughter strands are okay. The parental strand will these dimers will be then removed by excision repair. So this excision repair will come later and remove this, and these will be two thymine dimers will be added instead of these two thymine nucleotides will be added instead of this dimer. So if we look at a little more, let's look at a little more detailed version of homology directed repair. So here this is a double-stranded DNA. It's our DNA. There's a double-stranded break in this DNA. So first thing that happens is when there's double-stranded break, there will be 5' to 3' resection. That means the DNA will make these 3' overhangs, or single-stranded DNAs. These single-stranded DNAs are bound by these RPAs, or replication protein A. These replication protein A are the single-stranded DNA binding protein to avoid winding over to itself. The binding of RPA to exposed strands recruits these different proteins, especially RAD51 and RAD54. There's a combination of these proteins. Number of RAD proteins are recruited at the site. Then RAD51 is it's involved in looking for homology. It's a homology search protein, and it also helps in strand pairing. RAD54 is a helicase, so these two protein bind. These then recruit a RAD52. This RAD52 mediates the homology search along with RAD51. So here now we have this broken strand. This broken strand of chromatid will look for the homology in the sister chromatid. So these are sister chromatids. These open ends of DNA, these single-stranded pairs, these will look for homology wherever it finds the homology. So this one is looking for here, this one is looking for here. When it finds the homologous sequence on this homologous sequence on the template, it will synthesize that sequence. So here it will synthesize that sequence and fill the gap. Here it will synthesize the sequence, fills the gap, makes this holiday junction. We will talk about more of these holiday junction and this mechanism when we talk about DNA recombination. And then it leads to either non-crossover homologous repair or crossover homologous repair, which is more common is the crossover homologous repair, and this will lead to formation of these it will complete that sequence by using the information from sister chromatid on this chromosome. And you can see here, that's why it's really it can be used as a tool if you give enough homology to the sequence. So that's why when we are doing CRISPR with the homology directed repair, we also inject this plasmid with large homology arm of 2000 base pair, so that the sequence can find that homology and copy that sequence into the repair mechanism. Another mechanism of double-stranded break repair is called translation synthesis. This is also known as SOS repair or error-prone repair. This is really the cell's last chance effort when there are severe mutagenic conditions and the cell has two choices, either to die or fix whatever it can. So when it chooses to fix whatever it can, it's translation synthesis. In E. coli, excessive mutagenic activity activates the RAC-A protease, which activates UMUCD operon. UMUCD operon here is UMUCD operon example of this. This forms DNA pol 5. So remember, DNA pol 3 is the major replicative enzyme, the replicative pole in E. coli, but pol 5 can be used. Pol 5 is required. Let me start it again. In normal, no DNA damage, there is the LEX-A protein is the SOS operon is repressed because this LEX-A protein acts as a repressor. It binds to this LEX promoter and keeps it repressed. So that means it cannot activate the other portions of this SOS operon. So operon is repressed. But when the cell is distressed, so it has a distress signal coming, the DNA damage induces this RAC-A protease function. What does that mean is once the RAC-A protease is functional, it binds to this, inactivates the RAC-A. When the RAC-A is inactivated, this SOS DNA repair genes, which were repressed by LEX-A protein, are now free for expression. So these are now, because this protein is not bound to the promoter region, this repressor protein. Since the repressor is not bound, now gene is activated. So same thing here. There are now the different push parts of this operon. These are now free these are free or these are activated. So these together, these different portion of these genes will then translate different portion of pol 5, and this pol 5 will take over instead of pol 3, and this is called activated SOS operon. So in translation synthesis, DNA pol 5, which is a UMUC and UMUT, it performs the translation. What does that mean is this polymerase will ad-lib its way past a blockage. So using whatever DNTP it can find, it will just add those DNTPs. So here, this is DNA pol 3 as it's working here at the replication fork, is replicating. There is a thymine dimer. This thymine dimer, the DNA pol cannot pass through the thymine dimer, but pol 5 can. So DNA pol 3 will be now disassembled from here, and instead of DNA pol 3, pol 5 will come and bind to this area. and this pole 5 will add whatever it can at this portion. So for example, it had G and A here, it just added that G and A, so at least one of this pairing is correct or one is incorrect, and it will then it will dissociate once it moves past that damage, and pole 3 will come back and replication will occur as normal. And so since it adds any nucleotide that's available there's chances that a quarter, 1/4 of the time, the choice will be correct because there are 4 different nucleotides. So the chances of mutations are very high. In humans, in most higher eukaryotes, the repair mechanism is a little less error-prone because it inserts adenines opposite pyrimidine instead of ad-libbing its way. And since it adds adenine in most of the time, the dimers are thymine, so it's usually most of the chances are higher to be correct. So when there is a cytosine thymine dimer, then it will be incorrect. It will be CA pairing, but with T, the 2 T's, it's correct. Okay. In eukaryotes, the ubiquitination of sliding clamp, so here of pole 3, will recruit a translesion polymerase, which is your pole 5 equivalent of E. coli or UMUD2. See, that's what it's called in eukaryotes, so that will be recruited in place of the main pole delta. Non-homologous end joining, or NHEJ, it deals with double-stranded breaks as well, and this is the most common repair mechanism in higher eukaryotes. So most of the time when we're doing CRISPR, what we would see is non-homologous end joining comes first, or is more common strategy for the cell to fix these double-stranded breaks. And this is probably the same thing with the DNA repair as well. The non-homologous end joining is more common compared to homologous repair. And translation only happens when it's when there is just not much there to do. For NHEJ, or non-homologous end joining, there are a couple of different members of this pathway are super important or very important. Q17, Q80, for example, these proteins, they recognize broken ends. You can see here, this is a double-stranded break in the DNA. When a double-stranded break happens, these Q17, Q80 bind to both ends of the DNA, keep it from breaking more. And so this Q17, 80, then they recruit these protein kinases, and Artemis is also a protein kinase. This Artemis has 5 to 3' exonuclease activity. It also has an endonuclease activity, and these activities are activated by kinases. These PKCs, along with Q70 and 80, they process the available ends, and then a bunch of different proteins are recruited at this site. Protects and process the broken ends and then join them together. Exactly how it does is not very well understood, but what we understand so far is there are 7 components of NHEJ pathway. Q17, Q80 are the most fundamental components of non-homologous end joining. And in CRISPR experiments, when scientists want the cell to repair the DNA by homology-dependent repair or homology-directed repair instead of NHEJ, one of the way we achieve that higher HDR efficiency is by knocking down Q70 or Q80 at the same time when we are injecting the guide RNA and guide RNA, Cas9, and plasmid for homology repair. At the same time, if we also inject double-stranded RNA against Q70 or Q80, that will stop non-homologous end joining pathway and will lead to homology-dependent repair pathway or directed pathway. So Q70 and 80 recognize broken ends. They constitute a heterodimer. So here, this is a double-stranded break. Cu70, Cu80 make heterodimer, and these Cu17, 80, they bind to each end of the DNA. Now these DNA PKCs, these Artemis, including these PKCs, these come and bind. Since Artemis has all these exonuclease and endonuclease activities, and these activities are activated by these protein kinases here that they bind to. These all bind together. And then other proteins come and bind as well. They try to find if there is any homology. They can find. If there is no homology is found, it will just decide. The cell will decide whether to make to add more nucleotide or delete some of the nucleotide. And either way, there are a couple of different factors that will come and bind here as well, and the ligase will then fix whether it's deleted or added nucleotide. Ligase will fix the next. So again, it's error prone because there is no way for the cell to know what was going on here, whether there was a sequence similarity, whether there was any nucleotide here or not. Artemis is because it's a 5' to 3' exonuclease, and it is a latent endonuclease as well. It is activated by kinases, and it processes these available ends that it can, whatever way. This is an example of non-homologous end joining, how the mutation happened with non-homologous end joining. So here, this sequence is AACATTGT. If there is a break happens, it's a double-stranded break happens in this sequence, and it's try to ligate itself, try to find some sort of homology. What it found here is this A2 homology here, but this A2 homology is because it's a mispriming. This is not the correct A and T here. So what that would lead to now is because it's a misalignment, but it's to the cell it looks like it's an alignment. It will use its cell cycle dependent proteins to fill in this misalignment and make these extra nucleotides. These extra nucleotides are now going to frame shift, and that will change the protein. Same thing here. If it decides to instead of inserting, if it's decide to delete, then it will see this homology and the rest will be deleted. Rest of these nucleotides will be deleted, so that again it will lead to frame shift and it will lead to a different protein sequence. All of these events require Cu70 and 80 ligase 4 and these other XRCC4 dependent proteins. Microhomologies are these extremely short complementary regions like I just showed you in that non-homologous end joining, and these appear sufficient to bring single-stranded DNA together within a protein complex, and these can be ligated by ligase 4. However, this repair mechanism, it loses genetic information because it either deletes or inserts nucleotides, and therefore it's mutagenic. Site-directed mutagenesis is this is a lab technique. It can make specific hypothesis-driven mutations that can include insertions, deletions, transitions, and transversions. These most of the time we do site-directed mutagenesis to understand the function of a particular sequence or a particular nucleotide. How this will affect an amino acid. If we only want to change one amino acid, we can do those site-directed mutagenesis. If we can change multiple nucleotides as well, multiple amino acids as well, but this is but we want to do it a specific site where we are we're making these mutations in. There are two methods of site-directed mutagenesis, either a phage based or a PCR based. Both have the same basic strategy that we will talk about in the next slide. How it's carried out is a mutation is incorporated in a short single-stranded DNA that serves as a primer for replication, so it's either a phage or a PCR. This replication creates daughter strands that have the mutation, and once the daughter strand has a mutation, it can be purified from the unmutated templates. So here you can delete or you can insert piece of DNA, segment of DNA. So here this is the parental plasmid. In this parental plasmid we have these different regions. So you have all these different regions. This is the linear product. So what you do is, if you want to make this whole, if you do not want to make any changes, you will design the primer in a way that these primers just touch each other. So it will, this primer 2 will go in this direction, primer 1 will go in this direction, and it will replicate the whole circular DNA. But if we want to delete a portion of this plasmid, make site-directed mutagenesis, and we said, well, I want to understand what's the function of this particular sequence here. So I want to delete it and see what happens. So what you can do is you design your primer in a way that none of the primer overlaps or amplifies this portion. So your primer 1 goes away from this sequence, and primer 2 starts away from the sequence as well. So once the daughter cell, when it amplifies, it will only amplify from here to here, here to here. So this way you deleted that region, and then you can see, you can ask the question, what was the function of this particular sequence. Now, if you can insert a sequence as well, so what you do is you make your primer in a way that you have this extra region, extra sequence that you want to insert, and this sequence is overlapping in both primers. So that way, this primer making it this way, this primer is going this way, so it will also amplify this sequence as well. Now you have this sequence that's inserted inside the plasmid. So you can do these with the plasmid, you can do it with the phage, or you can do with this PCR as well. Okay, so far we talked about multiple different ways of DNA damage and repair. The DNA damage can happen in a number of different ways, and similarly the DNA can be repaired by number of different ways as well. We talked about single-stranded damage, and the DNA repair mechanisms for that is base excision repair, or BER, or nucleotide excision repair, or NER, or mismatch repair, MMR. We also talked about direct reversal of the DNA damage. These are photoreactivation and methyl group removal, examples of that. We also talked about how the double-stranded breaks in DNA need to be repaired, and we talked about non-homologous end joining, microhomology mediated end joining, which is a kind of part of non-homologous end joining anyway, homologous recombination, and SOS repair or translesion. So these are all different mechanisms, DNA repair mechanisms that are there, that now you know all of these. How DNA damage, the scientists have used the DNA damage for our own benefit. I think the sterile insect technique is one of the really good example of that. So we know that radiation leads to mutation, and this is something that we can use for these pest organisms, where what people do is they have these huge colonies of insects. They grow a lot of insects, and take the males and take the eggs and radiate them. And all these the sterile males are then identified from those pools of gamma radiated eggs, and these sterile males are then released into the wild. So these sterile male will mate with wild type females, and possibly mate them sterile as well, so there is no offspring. Males are usually a better choice than females, because males do not reproduce directly, do not lay eggs. So these can be thousands and hundreds of thousands of males can be added into the system, can be into the wild, and hopefully they will compete with the wild type males. The screwworm fly used to be very common in the US and pretty much all over the northern and central South America, but this has now been eradicated completely from US. There are some reports of screwworm fly coming back to Texas, but most of the part it has been eradicated, and the eradication was carried out by gamma radiations of eggs and then releasing sterile males. So this is screwworm fly. It's a pretty cute little fly, but this cute little fly, the immature forms, they make they have these mouth parts, these burrow inside the skin, and then they eat the flesh. So they're not very nice. And these were then these were eradicated by gamma radiation, but now there are new techniques of sterile insect techniques, more genetically modified organism that people are now using, especially for mosquito control, and some of the flies, especially in Australia, they've been melon fly and citrus fly that they're trying to control by eradication. Okay, so this is all for DNA damage and repair, and we will start the next module on Monday, and there will be a quiz on Monday as well.